it didn't occur to me that I was the one who was actually supposed to start it. So I'm sorry we're a little bit late. I knew I was going to introduce my favorite scientist, but let's all settle down. I can't see if you're settling down because I got these bright lights in my eyes. You all know who I am, Eileen Sullivan, and we're at the point now, which is pretty much my favorite part of any ed forum that I'm involved with that also involves Dr. Hunter. Because, and I told him that I was going to say this in my introduction, he is my favorite scientist. And fortunately, that's going to coincide with what he's going to talk about today. If it's the first time you've heard Dr. Hunter, or Zach, we'll just call him Zach, when he started working at Dana-Farber 20 whatever years ago, we called him a wunderkind because he was. You know, he wasn't even Dr. Hunter at that time. He was just Zach. And he was working in the lab. But thanks to Dr. Treon, I just learned this last year, I think from his mother, he was stolen from the math world and brought into the science world of WM. And thank goodness for that it happened, because he's been involved, and you can read his bio in the booklet and see all the things he's done, all the genetic discoveries that are because Dr. Hunter knows the right questions to ask. So from Dr. Soroshik and Dr. Gertz this morning, we heard a lot of results of science. We heard what they learn by doing their scientific research. But what we can learn from Dr. Hunter is, what are the questions that needed to be asked in order to get that research result? And so that's why I think that's what a true scientist's mind is. They're not just looking at the answers. They're looking for the questions. So science could not be more important for what we're doing, and science is why we're here where we are today.
Hey, thank you so much for inviting me here, and this is something I really look forward to, actually. For those of you who have heard me speak before, I often will choose a subject and do sort of a relatively deep dive on it and sort of talk about how it relates to Waldenstroms. But today we're going to do a little bit different, right? I really love the theme of this conference, right? We're in Florida, this is relaxed. And so instead what I'd like to do is really focus on the issues of, like, why science? Why do we do basic science work for WM, for cancer in general, and then bring some of those questions into WM towards the end. So to be clear, this talk is not about facts and figures. You do not need to remember anything about high school biology. We will cover a little bit at some point, but really don't get too hung up on the names or like details. If there's some statistic you're not worried, you don't understand, it's really okay. We're talking about big picture stuff. And so we're going to break this talk up into 3 basic parts. The first part is really trying to think like a scientist. Now some people might take that and feel a little bit like, I didn't sign up for this. But think of it instead of just being a problem solver. Everyone encounters problems in their lives where there's something you need to do and you're not quite sure how to do it and you have to think it through. So we're going to go over a couple of questions that are relevant to WM, and because you'd be surprised how often the issues that we are dealing with as researchers are actually, at least on the conceptual level, quite straightforward. Then we're going to talk a little bit about DNA and epigenetics. So some biology, but not too much, just very light. And that's to be able to transition over and talk about how this stuff plays out in Waldenstroms and what are some of the most recent findings. If you feel like this, again, remember, it's Florida, it's relaxing, big picture stuff. Don't worry about it, all right? So my name is Zachary Hunter. I work at the Bing Center for Waldenstrom's Macroglobulinemia at the Dana-Farber Cancer Institute. If you're ever in Boston, please come and visit. And so for this first part, we're going to again put on our scientist caps and think about some basic questions. And so the first thing, if you were a scientist trying to cure WM, is like preclinical testing, right? We want to develop drugs, right? New drugs are good, right? The better they work, the better, right? But it takes a huge amount of work. I mean, boy, like anyone who's ever worked with the FDA, like getting it through clinical trials, making sure it's safe for humans, making sure, you know, everything is going to be okay, it's stable, all that stuff is huge, right? So before you do this, you really want to make sure it works, right? I mean, you'd hate to do it and then find out, yeah, actually, it's perfectly safe. It just doesn't do anything, right? So a lot of what we do in the lab is test out new ideas. Hey, what if we target this gene? What if we use this protein? Here's this cool observation we made, right? So what's the problem with this? One thing that people don't realize is as hard as it is for us to remove all the Waldenstrom cells from your body, we cannot keep them alive outside of your body. Yeah, think about that, right? So this poses a real problem for us, right? We take them out, they're already starting to die. So when we treat them, there's two issues. One, they're already dying, and that's going to cause, you can still do it, it's still very useful, but it's already causing some signal issues, right? And they're not getting what they need. So there's going to be some drugs they're going to respond to really, really well in that situation that they could care less about if they were getting their full support in the bone marrow, right? So what can we do about this? Well, cell lines. So one of the things that people have worked very hard over many years to do is develop samples derived from Waldenstrom's patients that can grow continuously in culture, right? So we can actually just basically come up with a broth that they'll grow in, and we just have to keep changing them out. It's kind of like keeping a pet. We do have to have people come in on weekends to, like, you know, pet them, clean the cage. And that's great, right? So we can do all sorts of tests. We can change their genetics. We can introduce new things. The problem is that they grow really well in culture, right? Which Waldenstrom cells don't do. Right? So that means that we have an issue where any drug that's going to target one of these areas that Waldenstrom's depends on in the body probably aren't going to work. And so a good example of this would be the BTK inhibitors, like ibrutinib and zanubrutinib. In our WM cell lines, they don't really work. If this was our only way of assessing what was going on, yeah, we probably would have taken a pass on those. So it's an issue, right? Even so. So we can sort of go and come up with other ways of getting at this. We can understand the pathways well enough to look and make sure they're being inhibited if we really think it's going to work in the person. We can also grow them in mice, the cell lines in mice, and that sort of simulates being in the body. Sometimes that kind of helps give us a sense, but still, it's a poor substitute. So this is a real issue. This is something that basically all Waldenstrom's researchers and all cancer researchers deal with all the time. So the other thing I'm going to do as we go through this talk is, and this is something I think is really important for people who are here at this conference, especially those of you who are here for the first time, is what an unbelievably important role the IWMF has played in changing the research landscape of Waldenstrom's. And so a lot of people I'm going to be highlighting periodically are actually people who have received IWMF grants, right? And also I like the idea that there's all these people who you probably don't know who are working on your behalf all the time. So I'd like you to meet just a few of them. So Erna Magnusdóttir is from Iceland. She's actually working to grow out artificial bone marrows, right? So we can take real live Waldenstrom cells from patients and grow them there for testing, and then to understand those interactions between the bone marrow and the microenvironment. We're working together on this. She's amazing. Shrong Liu is actually from our group. And if you ever hear Steve Treon, who does a lot of drug development actually, ever hear him talk? Well, Shrong's the guy who actually did the work and organized those studies, put it all together. He's been really, really amazing and actually just published a paper introducing a new cell line, BCWM2. And then Constantine Mitsiadis, I promise they're not all going to be from the Dana-Farber, just this slide. He's working with cell lines to knock out genes and to understand what genes cell lines are dependent on. And then we can go back to Waldenstrom samples and try to see if we can find new drugs, new targets. And actually, he just got renewed for a new roadmap, and we're all really excited. We're collaborating with him on this, and hopefully we'll have some new results next year. So that's another thing, Let's go on to the second question here. And this is another simple question, right? Things that you don't think of as being all that important. I mean, you think it's interesting, but maybe not super important. How long is it going to take before someone needs therapy? Right? Interesting problem is we typically don't really know. I mean, it's not that we don't know what progressive disease looks like. Once you're sort of heading on that track, we can say, hey, you're heading on that track. And you usually don't need us to tell you, right? You probably know. But then the real question is if you're not on that track, right? How do we know in 3 weeks you won't be on that track? Or 5 years? Or some people I've seen go 20 years, right? And are there biological differences? Are things changing during this time? I mean, this is a real question, right? We talk about doing early therapy, early interventions. Well, is the biology the same early on than it is later on? And if we can't easily determine who is actually are about to transform into more aggressive Waldenstroms and who's just going to stay and be asymptomatic for a long time, it'll be really hard for us to figure out if there's any differences, and even just figuring out which people we're going to do early intervention on. It's complicated. Even things like bone marrow are predictive, but they're not actually a perfect correlate. There's people with advanced disease with small amounts of marrow and people who stay perfectly stable with fairly high levels for many, many years. So there are some clinical tools, including the WM asymptomatic score, which is available online actually, and that's relatively straightforward to use, that can do some basic predictions. And one of the big breakthroughs that came out of our group was the development of a computational score, which is not something that we can do, unfortunately, clinically yet, but we can do on the samples that we're studying, that will allow us basically to predict this and to line all the samples up on a spectrum from sort of smoldering Waldenstroms to active disease. I'm going to give you some examples about why this winds up mattering in research. So let's take the case of CXCR4. It's a mutated gene. It's pretty frequent in Waldenstroms. I think a number of you may have heard about this or be familiar with it. And what happens when it's mutated is it's turned on all the time. And what it does is it causes cells, surprise, surprise, to migrate to the bone marrow. This probably sounds familiar. So, not surprisingly perhaps, people, as you know, sometimes have lymph node involvement, sometimes they have spleen involvement. You'd think, well, hey, if they have this mutation, they're more likely not to go there, they're more likely to go to the bone marrow. And that's exactly what we see with lymph node involvement. Very significant, very predictable. We almost see it with spleen involvement, but it's always just shy of being statistically significant. So what's wrong, right? Is there something wrong with our conception about how CXCR4 is working? Well, the real issue is that unlike the lymph node involvement, not everyone's equally at risk for spleen involvement. Actually, it's really something that just happens at sort of towards the more advanced disease. So if we really want to understand what CXCR4 is doing in this context, we need to be able to stratify people and figure out who is at risk for having splenomegaly in the first place. And that means we need to be able to line these patients up. And if we do, there it is. CXCR4 is really quite important. Now, for those of you who are looking at this and being like, that's okay. That's totally fine. So there are statistical tests, and we don't need to go into the details, where we can test multiple factors simultaneously and test their contributions to some kind of result. In this case, whether or not someone has an enlarged spleen. And what those boxes are is our estimate as how it increases or decreases the odds. Everything to the left of the dashed line is decreasing it, everything on the other side is increasing it. And those little bars are just telling you, if we were to do this 100 times, right, 95 out of those 100 times, it would be within the bars, right, if we were to repeat this exact same study. So basically, if those bars aren't touching the line, it's probably very significant. And so you can see the evolutionary score, the E score here, which we'll talk about later on, this actually is highly, highly, highly predictive of whether or not you're at risk for splenomegaly, and CXCR4 decreases that risk, right? But we can only see it when we have the two things together. What's another example? So there's a lot of variations in normal human biology. And this is, again, an issue. So when we go and we look at bone marrows, one of the things we had noted for a long time is sometimes we see lots of cells with mast cells around them, and other times we see a lot of cells with myeloid cells around them. Two different cell types, you don't need to worry about the details. But they're very, very different from each other. And this is interesting. We don't really, it's hard to know. And one of the things that can happen is you can be like, oh, I think I see a pattern, but for all we know, it may be one group of people recovering from a cold, or recently had COVID, or I don't know, maybe one group is vegan and the other one is like eating like tons of meat every day. Who knows, right? There's a lot of normal human variation that comes up with lots of normal variation in all across your body. And a lot of it doesn't really matter. But as it turned out, once we were able to line up the samples from very early to very late, we could actually see it as a steady progression from lots of myeloid involvement and few Waldenstrom cells to more Waldenstrom cells and a lot more mast cells, and the myeloid cells slowly disappear. And so this is something that was only possible to see because we could line them up. So this is the kind of thing that, again, would have been impossible to see without this sort of basic research backing, even though, technically, we've seen thousands and thousands of these slides. So Mark Bustros, another Waldenstrom's researcher, he actually developed the asymptomatic Waldenstrom score. Romanos does single cell work and has done really some very cool stuff about the progression of Waldenstroms from MGUS. And Philip is actually the guy who works with Carrasco, who made that slide just in back, and is, again, also an IWMF recipient working on a mouse model with sort of unique Waldenstrom's genetics. So this is the last one we're going to do. And this is kind of going back to Irene's question, or Arlene's question, which is, how do we know what the right questions are to ask, right? If you know there's a mutation, that's great. We can say, hey, do you have MYD88? Yes or no. Do you have CXCR4? Yes or no. Let's see if there's a difference, if this impacts what we're studying. But what if there's something we don't know, right? This becomes a problem. So we can just throw drugs, for instance, at cells, at Waldenstrom cells writ large, and just be like, hey, let's see if it works. And what happens, especially when we're working with primary cells, is half the time it works and half the time it doesn't work, and we have no idea why, right? And again, sometimes it's just differences in general biology, but sometimes there's a hidden factor we're not aware of. And so this is a huge problem in data science in general, and there's a whole bunch of tools that we can use to try to come up with ways of either visualizing the data or splitting things on hidden factors to let us understand what's going on. But this is something that really is very, very active, and I'm going to try to walk you through a couple of problems of this. So this actually goes back to my master's thesis. Nobody has ever seen this, I promise you. Like the 4 people who had to review it and probably nobody else. But this is way back before we knew about CXCR4 or MYD88. We were looking at gene expression in Waldenstrom's. And there were these genes that were really overexpressed, mostly, right? Like 2/3 of people, maybe half were overexpressed, the other half weren't. But this was really exciting. No one had ever seen these genes before. I was also very young, so I was very, very excited. And so, I did what any good cancer biologist does, is he's like, okay, well, how do we smash these things to see if we can cure Waldenstrom's? This would be great, right? And then we ran into exactly this problem. The cell lines that we had didn't express them, so we couldn't use those. When we tried to smash the primary cells, some of them seemed to work, but some of them didn't, and we didn't really understand why. So luckily, right after this, I wound up focusing the thesis on something else because I couldn't get this to work. We discovered MYD88 and CXCR4. And boy, whoa, Waldenstrom's wasn't one thing, it was multiple things. And it was really different based on the different kinds of mutations that people had, right? If you had MYD88 and CXCR4, you had one level of expression, that's in green. If you just had MYD88 and no CXCR4, that's purple. Really, really different. Really, really confusing. Why would it go up with MYD88 and back down with CXCR4? That's an excellent question. We didn't have any smart answers. But hey, at least we could explain it. But there were still problems, right? when we looked at it more closely, there were outliers, right? And being an outlier, let's say on Wink2, shown here in blue and green, made you an outlier pretty much in all the genes we looked at, right? Almost as if they didn't really belong in that group. And it's actually exactly this kind of questioning that led us to discover the subtypes of Waldenstroms. This is something, again, we're going to talk about more towards the end, but I just want to bring up quickly now. But we found 3 distinct subtypes of Waldenstroms. And without going into too many details, basically there's an early form of Waldenstroms that everyone kind of initially goes through, and then they diverge out into 2 different distinct subtypes. And it's worth mentioning that one of them is about 80% CXCR4 mutated, and the other one is just about 7% mutated. So now let's go back to our original question, right? Here's the same data, split on CXCR4, colored by the subtypes. And you can see right away that CXCR4 has nothing to do with this, right? The subtypes are really determining what's going on here. And so what's fun is, in my master's thesis, we gave up on this project. But we actually published a paper in GPR1 just last year. We're about to submit a paper on Wink2, really any day now. We're about to launch into a big study on DUSP22, and we can do this because we understand the context now, right? We understand when they're up, why they're up, what those contexts are, and that allows us to understand their function, to study them, and to see whether we can derive new ways of treating and understanding Waldenstroms. But again, you can only ask questions if you know that there's something there to ask, right? So this is a big issue. A lot of what we work on in basic research is trying to discover what are the things that we're not asking. And I also just want to emphasize that this is actually shown to really matter. It's not just some thing that makes it look nice in the books, pretty pictures, as Steve likes to say. With bioinformatics, you got lots of colors and dots and things flying all over the place. It's really fun. But it actually really matters. So these are all examples of how drugs, or how different ideas that we've discovered in basic research impacts the response to different therapies. CXCR4 and ibrutinib, the subtypes as proteasome inhibitors, and we talked about the E-score. It turns out the people in the most advanced E-score really showed different biology, and because of this, we see different responses, again, with proteasome inhibitors and BTK inhibitors. And I'm not the only person working on this. There's actually a whole crew of people who are all awesome, and all, again, supported by IWMF grants. Patricia Mondello, who I'm actually working with right now on the first enhanced roadmap, bridging our 2 models of Waldenstroms. Gareth Morgan at NYU, and Tita Bagratoni in Greece, all of whom are doing different versions of the same thing. So I'm going to transition now and just talk a little bit about basic science, but not too much. Again, high level, don't get too worried. Genomics is very easy. I promise. All right.
He says that every year.
It's true, I do. So DNA, we've all heard about, right? But it's really straightforward. It's made up of 4 different sort of molecules that we call bases, right? And we abbreviate them A, T, C, and G. And that's it. We have about 3 billion of them. They're in a long string, right? And they're broken up into 23 chunks, right? And those big chunks are called chromosomes. And that's really it. The special thing about DNA is that it has a sort of complementary structure. So by design, the G's can bind to the C's, and the A's can bind to the T's. And this means you can have 2 separate strands of DNA which are perfectly complementary to each other. And what's great about this is, this is the real world. This isn't a textbook, right? There's reactive oxygen species, ionizing radiation. There's all sorts of crazy stuff that's going on in your body all the time, right? Damaging this. But, hey, if you have complementary copies, your C gets messed up. Your body looks like, this is messed up. Hey, what's supposed to be here? Let's look on the other side and find out. And you can repair it. It's great. And it's super stable. This is why we can go and we can talk about whether or not some Egyptian pharaoh died of a particular genetic disease or not, is because the DNA is that stable. It's amazing. So the problem, though, is 3 billion base pairs is pretty big. You actually get a full copy from mom and a full copy from dad. But you want to be using it, right? It's used to run your cells. And you can't be taking this big, massive thing and running all over the cells, all over the place. Hey, you got to go over here. No, wait, this thing has to go over here. That would be nuts, right? Logistically, anyone who's ever worked on a construction site or something like that, you can't have one gigantic thing that has to be 10 places at the same time. So you don't do that, right? You take copies of it, little tiny copies of just the section that you need, right? And you transcribe that off, and you send that off to where it needs to go. You don't need the whole thing, you just need the piece that you want. And that's called a gene. So a gene is just a chunk of DNA that can get used, that gets copied off, and sent out as instructions to the cell. That's the whole thing. That's cellular biology. Everything else is just a consequence, right? And we're going to talk a lot about how this gets more complicated over time. But this is the basic setup, all right? But here's the thing, right? With 3 billion pieces, things can get really complicated. And so, there's been a lot of news with AI recently and how amazing it is. And it is truly amazing, right? So 2 things to keep in mind. Computers only have 2 bases, true and false, or 1 and 0, that they work with. But here's an interesting thought. And I really want you to think about this, right? Which is things like ChatGPT will get trained with 10 trillion words. That's a lot. That's a lot of data, right? And it still doesn't understand what it's doing on some fundamental level. It's still basically fancy autocomplete. Impressive, really, really impressive, right? But it really has a lot of issues. So even though it's so big, what is the complexity in our much smaller, albeit more diverse genome and our DNA that allows for the complexity that we know we have, that the computers are still struggling to catch up on? That's really what I want to focus on for the next little period of time. So the first thing to understand is, just to review, is we have DNA, gets transcribed into RNA. These are the messages coming off the genes. And one of the main things that RNA does is make protein. You've heard about protein. Protein is what does all the work in the cells, right? And an important thing, of course, is that that protein can go back and bind to your DNA and turn on different genes to respond to your environment. That's awesome. But you should feel dissatisfied with that explanation. If you stop and you really think about it, put your scientist hat on for a minute, that should feel incomplete. Okay? And to explain where that sense of incompleteness comes from, I would like to talk about visiting kids' soccer games. So anyone who's ever been to a junior soccer game, right? You have the situation where all the kids are out there and they all see the same thing. They all see where the ball is, and they just make a beeline for it, right? It's just one gigantic scrum, and it's amazing there aren't more injuries. But that's not how professional soccer is played, right? In professional soccer, everyone has the same information. Everyone still sees the ball, but everyone understands their role, and they respond accordingly. And if you think about what's going on in the body, right, you have the same DNA. It's getting the same signals, but your body responds really, really differently, just like the pro soccer players. They don't act like kids, right? So there's something that's allowing your cells, despite having the same DNA, the same everything, to sort of tailor their responses and become specialized. And this is epigenetics. And so what turns out is that big chunks of your genome are wrapped up really, really tight. So tight that, in fact, they can't be used. Now, some of these can be unwound really fast. So it's like, oh, hey, I need this. Let's get this available. But some of them are pretty much locked down, right? And that means that each of your cells are effectively seeing a different genome, because you only get to see some subset of the overall genes. And this really allows us to have a much more complete picture of what's actually happening. This complicated dance between the genome, the protein, and the epigenetics. And so each tissue that you have, right, whether they're cells in your brain, your neurons, your blood, your muscles, your skin, each one is seeing an effectively different genome because either chemical annotations have silenced those genes or they've been wound up so tight they can't be seen. So you can give the exact same stimulus to each of these different cells, and each one will respond according to their type. It's a brilliant system. Really makes it so much harder to work with. And as you can imagine, of course, this can go wrong in cancer too. And so now I'd like to transition, now we sort of set the stage a little bit, to what's actually happening in Waldenstrom's. What's going on? So to start, a lot of the data I'm going to be showing you is from our, I should say, IWMF-funded study looking at nearly 300. Actually, the whole study is over 300. The subgroup we're going to be talking about is 249 untreated patients with Waldenstrom's. And they were all seen at the Bing Center, and we've been following these people typically for at least 8 to 9 years. So lots and lots of follow-up. We know what happens to these people. We can go back in time and sort of think about how did their genetics and what was happening at the time correlate with actually what happened much later on. As you can see, these are pretty typical of what we'd think of when we think of Waldenstrom's patients who have yet to be treated. So the first thing we discovered were these subtypes, right, which we touched on just briefly. And the thing I would like to highlight here is just how different those top two look. Now, I totally get it. Most of you probably don't look at these every day, right? So these are called heat maps. And the reason they're called heat maps is because of the way they look. Kind of looks like some parts are hot, some parts are cold. So the hot parts represent, in this case, genes that are really well expressed. And the blue parts are the same gene that's less expressed. And so when we see this kind of a pattern, we're showing a whole block of genes which are being turned on in one group and off in the other group, and vice versa. And let's contrast that with early WM, where everybody's on, but not as strong. And so when we first saw this, we were really confused. I'm not going to lie. But then we noticed that the early WM people were largely asymptomatic, largely smoldering Waldenstrom's, not 100%, but largely, and very statistically significantly. And that made us start to think, well, maybe this is like a precursor thing. So we looked, and we could see how long it took people to get treated. So how many years it took before someone received therapy. And sure enough, all those people who had this sort of early WM, fall in this early WM group, all of them took a lot longer than the people who were in the first two groups. It was the memory B cell-like and the plasma cell-like group. And we can use different techniques to visualize the data. And so in this case, we're sort of, each dot here represents a patient in a pretend three-dimensional space. But what you can see here is it almost sort of looks like a U shape. And what's at the bottom of that U, in the center, is mostly this early WM group. And as you go off to either side, you get your subtypes. The the B cell-like, and your plasma cell-like group. And the interesting question is, what is going up and down? Right? What's the vertical element of this? It turns out that's a third factor that we're going to talk about in a few minutes, which is this WM evolutionary score, the thing that allows us to track how far along in progression people are from like smoldering Waldenstroms to symptomatic disease in need of treatment. And so you can really see this evolutionary, diverging evolutionary paths. And there's some interesting clinical differences too. So we mentioned in the B cell-like group, 80% of them are CXCR4 mutated versus just 7% in PCL. That's true of some of the other genes as well, like CD79B, which is 9% versus 3%. And in plasma cell-like, we see an enrichment for mutations in the Notch pathway. But perhaps more relevantly, we see a lot of deletions in 6q, 46% versus 28%. But going back to this idea that sometimes it's important to keep an open mind about these questions. Not everything is necessarily the same. We've, for a long time, been treating 6q deletions. So this is a section of one of your chromosomes that can get deleted in Waldenstroms. That just means one of the copies goes missing, gets taken out and thrown away for reasons we actually don't really know why, but does happen in cancer. We just assume they're basically all the same. Yeah, there's some differences, but again, normal biological diversity, there's some target that they're trying to get at. But when we actually split it by subtypes, the first thing we noticed was that one of them was losing a huge chunk of the chromosome. And the other one was just losing these little tiny parts. The same little tiny part, interestingly, but just that one little tiny part. They really weren't equivalent to each other at all. So again, going from, oh, hey, some people are like this to some people are like that, to all of a sudden being able to explain this diversity, and now we know to study them differently and interpret them differently. So what causes these subtypes? You knew we had to loop back around to epigenetics at some point, right? So let's think about this idea. We were talking about epigenetics in the context of different tissues in your body, right? Why your skin and your heart and your eyes all are different, even though they have the same DNA. Well, the same can be true for cancer. As cells develop, as they move from a less differentiated cell, like a stem cell, into something that is more useful, actually does some function in the body, it kind of goes through different stages. And those stages are controlled, again, by unwinding or hiding individual genes. So each stage has a slightly different set of genes. So let's say we were to introduce something like a MYD88 mutation or a CXCR4 mutation at two different stages of the development. It's the same signal, right? But they see slightly different genes. They have slightly different tools available to them and will respond a little bit differently. And this is called the cell of origin theory of cancer. And one of the interesting things about this is when you look at the epigenetics of cancer, the chemical annotation, which we only touched on a little bit, but you can chemically modify DNA to make it harder to interact with. Most of it, the cancer cells don't really care about, to be honest. It just doesn't really matter. Some of it really matters and gets changed, but most of it doesn't. Most of it is just left over from whatever the cell thought it was when it first became cancerous. And it's just left alone, because why bother, right? Like, basically, this describes my basement, right? Like, things I put in there, I'm going to get to it someday, it just kind of sits there, accumulates. Nothing ever moves. Same idea. So we can measure all these different chemical annotations in Waldenstroms and kind of group samples a little bit and cluster them by similarity. So similar samples cluster together. And the first thing you notice is there's two clear clusters there on the bottom, right? Turns out that's your plasma cell-like and your B cell-like Waldenstroms. The B cell-like completely overlapping with healthy donor memory B cells. The plasma cell-like group, co-clustering, interestingly not with healthy donor plasma cells. That's that group up there in green on the top, but with IgM multiple myeloma. Very different disease, Very, very different disease, very different genetics. But it does look like it has a very similar cell of origin. So this brings us to the second thing, which is looking at this idea of the WM evolutionary score. And again, this idea of using different visualizations to understand what's going on can be helpful. And even then, it can take us a long time to fully wrap our heads around what we're observing. But what you can see, I particularly like this method. It's not that it's important, it's called diffusion pseudotime, or DPT. And what you can see is on one side, everyone's sort of clustered together. And as you sort of move forward, it follows this arc. And as that arc goes forward, it splits cleanly into two groups. And those two groups just happen to correspond to our two subtypes. And the other thing I actually forgot to mention in the previous slide is when we looked at the early WM slides, they were already fully co-clustered with either PCL or BCL. Again, indicating that early WM is really a shared state, if you will, of early WM. All early WM looks the same, but the cell of origin is still the cell of origin. It's still distinct. And what kind of Waldenstrom's it's going to become has already been determined. And you can really see that here too. The other thing that we were able to show, because when we look at this E-score and as it sort of evolves and as they split off in these two groups, is we can track and show that the average number of mutations we can observe in the samples is steadily ticking up. And the average bone marrow is steadily ticking up. The rate of symptomatic disease is steadily ticking up. But the most interesting part about it is when we go from time of biopsy to time of first therapy, it's very, very predictive. But no, it's not predictive of time from diagnosis. Because one of the questions we have to ask is this just, maybe we're just looking at more aggressive Waldenstrom's, people who just rush the process real fast. And that's not the case. There's no real association here. This is really a measurement predicting from the time that we're measuring to therapy. And this is important. Again, it's interesting to know. Obviously, everyone likes to have predictions, but it means that we can start to study the biology for the first time because we can sort the cells. We don't have to go back, wait 10 years, find out what happened, go and make some guesses about where they were in their development based on how long it took for them to get therapy. This is really, really important because this is a real difficult thing for us. Keep in mind how long it can take for WM patients to evolve and need therapy. We'd be doing this study for the next 10, 20 years. I mean, I would be fully retired. So, if it's so useful for biology, what's happening? Right? That's a good question. So, it turns out the answer is really strange. And so we're going to sort of talk about it in two different phases. The first thing, and this is very counterintuitive, but asymptomatic Waldenstrom's is producing a lot of inflammation, like a lot of inflammation. And it's sending out signals to try to pull the immune system to itself. Now, you might think, and we thought the same thing, right? Why, if you're a cancer cell and the immune system is actively hunting you, would you go around and gear them up and then be like, hey, I'm right here. You should totally come here, right? That seems like a really weird move. And it is a little bit. But there's two things to keep in mind. One is that the kind of inflammatory signal that they're producing is something that can actually be received by something called the toll-like receptor system. Now, no need to know this, but this is the system that MYD88 is the adapter for. So this is on pathway. The cells are secreting something that will activate the pathway that MYD88 is on. CXCL12, one of those genes that's so high early on that helps bring the immune cells to it, that binds to CXCR4. right? So the cells are secreting things that they're receiving. So that's one part of it. The other part of it is you have to put on your regular biology hat for a minute. What if you have chronic inflammation? What if there's something wrong with your body where you have a chronic stimulus that's happening all the time? Your body can spend the rest of its life freaking out about it, but that's not helpful. It's probably damaging. So your body's evolved ways of seeing that situation and being like, okay, cool, we get it, this is a bit much. We're going to just locally kind of dampen this down, right? And it turns out that this is a really common cancer tactic. If you were to go and take these same genes and ask, what are the things that cause your T cells, for instance, which are cells that go out and look for things like cancer cells, to become exhausted and stop functioning, it's these genes. Chronic stimulation with these genes will get you there. So what is it doing? It's creating this exhausted, suppressive environment, perfect for the Waldenstrom cells to grow out. And so this is my final researcher highlight, but I'd like to start with Steve Ansel, because I know all of you have probably seen him around. He does a lot of work for the IWMF, but he also, it turns out, has been working in exactly this space of cell-cell communication, and actually did a really nice paper with Patricia Mondello, who we talked about earlier, on exactly this phenomenon in Waldenstroms. Maria Luisa Guerrera works with me and is an amazing researcher, and she's been working on Wink2, and it turns out Wink2 is heavily involved in this process. And again, we're actually about to publish a paper on this, and she's been the recipient of the Kyle Award and a seed grant from the IWMF. And Hao Sun from our group does single cell work. Again, mostly right now studying immune responses in response to ibrutinib, but has done some really, really fascinating work on this, and again, is just a newly minted Robert A. Kyle Award, and we're all very, very, very proud of him. So what are the other set of changes that are happening? So this is where it starts to get a little bit more confusing, and I realize it's not been straightforward. But there are genes, if you've probably heard about Waldenstroms being stuck somewhere between a B cell and a plasma cell, right? About stuck halfway in between. Well, what's really weird is that the early smoldering forms of Waldenstroms look a little bit more on the plasma cell side. Not plasma cells, but pushing in that direction. And the more sort of mature symptomatic Waldenstroms looks a little bit more on the B cell side. And we can see that in this data. But what's really fascinating about this is that the biology is changing. We're seeing a shift in the biology that is much more towards a B cell-like signaling complex. So things that B cells are really good at doing that plasma cells really don't do. And again, it's doing this in a way that is right on the MYD88 pathway. Right? You can almost think of it as like you have a car engine, and you start it off with like, I don't know, one of these old beat up jalopies you get at on a discount from a friend of yours, and you're not totally sure if it's going to work, right? And you're like, okay, fine. And then you rip out the engine, you completely redo it. The outside still looks like trash, but now it's got a Ferrari engine in there, right? And I think that that's basically what we're observing. It's not really that they're changing. They're still the cell types that they are. But their internals are shifting in a way that's really allowing them to maximize the mutations that they have and the signaling that's already there. And that's why you start to see them grow out. And that's not the only thing. We talked about the idea of a shared state on early Waldenstroms. And that's a weird thing to think about, right? They start off different, they end different, but somewhere in the middle they look the same. That's, again, sort of a weird statement. And I'm not going to claim that we fully understand this. But when we started looking at the E-score, we noticed some things that gave us some clues. And there are genes, we talked again about epigenetics, that are specific to different cell types. And as biologists, we can use them to characterize different cells in your body. It's like, oh, hey, I see that protein. I know what that cell is. I don't even have to see it. That's a T cell. Oh, that's a myeloid cell. That right there, that's a neutrophil. I got it. Cool, you don't tell me anymore, I got it. Unless it's cancer. Because let's look at CD4. This is a list starting with hematopoietic stem cells. It's a fancy word for saying cells that know how to make all the different kinds of blood cells in your body. Waldenstrom is right there, halfway between this like B lymphocyte and a plasma cell. But where's CD4? CD4 belongs on T cells, right? Different branch. RAG1, RAG2, IGL1, that's not supposed to be there. This is a very temporary thing that's very brief sort of flash of expression as you're trying to generate your B lymphocytes. CD33 is on a completely different branch. It's a myeloid gene. CD34, those are stem cell genes, right? Why are your early Waldenstroms expressing all this stuff? So what we think is happening is instead of this sort of normal progression, where you go from precursor cells to pre-B cells, to germinal center B cells, to memory B cells, to plasma cells, roughly. I'm simplifying things. We think we have these generators, so precursor cells. We think maybe MGUS. And then, something terrible happens. And part of that something terrible, we think involves not the cells really becoming stem cells. They're not stem cells. And actually, when you look at the details of what the cell is doing, the cells very much think that they're memory B cells or whatever the heck IgM multiple myeloma is. They're very clear on this. What they're expressing, their epigenomics, that's what they think they are. But all of a sudden, they start reactivating stem cell genes. Actually, their chromatin has been radically opened up. Things they're not supposed to see, they're seeing all over the place. So even though the signaling hasn't changed that much, all of a sudden that same signal can do all sorts of things it never could do before. And that's what we think leads to all this weird expression. And that's also what allows it to sort of re-engineer itself, ultimately optimize itself, right? And then over time, it regains control of its chromatin, but just the parts it doesn't care about. And that leads us to our two cell types. So when we put it all together, we really have two different things. We have this early Waldenstroms, and it can either migrate out towards BCL or migrate out towards PCL, the two different subtypes. And we have this E-score, and this allows us to understand the changes that happen moving from early Waldenstroms, asymptomatic, smoldering, into aggressive disease. And for the first time, again, because we can do these things, we can start to understand the biology, understand the impact on therapy, and try to figure out what we can do about all this. And all this was possible through the support of you all. So thank you very, very much. I'd also like to thank WMFC, the Tim and Ginny Bless Investigatorship for Waldenstroms, of which I'm a recipient. Many of our donors, the Schul Foundation, the Orzag group, and all of our collaborators, but most of all the patients. This would have been completely impossible without you. I'd also like to thank everyone at the Bing Center and who's in the amazing team we have here. We all work together. And again, if you're ever in Boston, please come. We can give you a tour of the lab.